During the biological experimentation and the processes, the antibodies which are prepared, they have to be kept alive and they have to be kept active. Now, in this module, we will discuss that how it is possible to keep those cells active and how to store them for future, and that is called freezing the cells and the cell banking. That how we can prepare a cell bank which can be used later whenever we desire. Now, cell lines, they are very important for biologists to work with. And the cell lines are how to keep them for a long time and how to keep them active so that they are available for studies. So for this, to achieve this, the cell lines, they are taken, they are frozen to provide a source of inoculum for future culture. That is, for our future experimentation, those cells are taken, they are frozen, and they are used time and again whenever desired. Now, cells which are stored at low temperature, they achieve low temperatures, they are achieved using liquid nitrogen vapor and at stable for many years. That is through a branch of science which is known as the cryogenics. These cells, they are frozen, their biochemical activities are frozen by the liquid nitrogen vapors, and they are stable for many years to use for further experimentation. Now, the cells are transferred, it is done that the cells are transferred into specialized medium prior to freezing to protect them both when the temperature is lowered and also the temperature is raised, that is when it is thawed. Imagine the frozen cells, they are taken and first they are put into a special media prior to freezing. Now, this is very important so that if we don't do this, then we will damage the cell structure which is there, and the cells, we won't be able to utilize them again after time. So during freezing, there are two processes. One is putting them to a specialized media, then to put them or to freeze them at very low temperature, and again, while using them, we have to revive them, and we have to revive them by raising the temperature so that they are thawed, they are melted, and they are used for this purpose. Now, serum containing, how it is done? That the serum containing 10% DMSO, dimethyl sulfonyl, that works well as a freezing medium. So this DMSO is a very interesting chemical. It is used in the cryogenic experiments, and it is anionic as well as the cationic, and it organic solvent. It is repeatedly used as a cryogenic media for freezing the cells. Now, a typical freezing that should contain around 1 million cells. That is, when we freeze, there should be around 1 million cells which should be present in there per mil so that they are frozen. Then the cells are harvested from the flask by tapping to dislodge them. Because the cell, when they are growing, they are adhering, they are sticking around the flask and we have to remove them. We have to remove them by just tapping the flask to dislodge them, so that we tap the flask walls and they are dislodged. Then, once they are dislodged, they are in solution, they are pelleted by centrifugation. We subject them to centrifugation, where they make a pellet and the culture medium that is removed from them. Now, after we get the pellet, these cells, they are resuspended. Again, they are redissolved, resuspended in 1 mil freezing medium, and that is chilled to 4 degrees centigrade, placed into a cryogenic vial, and transferred to a cell freezing container. That is, we have got a cell freezing container where there is a liquid nitrogen and liquid nitrogen vapor, and that vial is transferred into that container to make a bank of the cells. Now the freezing containers, what they are? Freezing container, they contain butane 1-ol, which when placed into 70 degree centigrade freezer, it controls the rate of freezing to 1 degree centigrade per minute. That is the control of the temperature, that is freezing 1 degree centigrade per minute. It controls the rate of freezing. It is not that it is immediately frozen and the whole cell structure, it is destroyed. The cells are left for minimum of 24 hours and up to 72 hours prior to transfer into a cryogenic state. So you see, we put the first to make them freeze, and then we transfer them to a cryogenic store, and we transfer it to a liquid nitrogen storage, which is cryogenic, very low temperature. And when we transfer it to liquid nitrogen, what should be? It should be rapid to prevent thawing of the cell. That is, we take from that medium, we take from that container, and then we put them into the nitrogen storage cryogenic cylinder, which contains liquid nitrogen. And this process should be very quick. It is not that we took those cells in the morning and then in the afternoon you put it in the liquid nitrogen. No, because they will thaw and they will use that. So cell banks, they are established from known positive clones and are produced in a way that maximizes the reproducibility between the frozen cell stocks and minimizes the risk of cellular change. That is called the cell banking. In this case, there is a depiction of how the cell banks works, that we get the cell and those clones which are used, they are stored there for making the cell clone. A positive clone, which is derived from known positive clone, is rapidly expanded in the tissue culture and can be kept for long time. So this is called a master cell bank and is stored at minus 196 degrees under liquid nitrogen vapors. This strategy of cell banking, it ensures that all of the vials of the working cell, they are identical and they can be used for further experimentation in the biological field.